Well, good evening, everyone, and good afternoon for those of you on the West Coast. I'm Dr. Joe Purita. I'm an orthopedic surgeon, board certified in orthopedics. I've been doing regenerative medicine for about the last 15 or so years. And within the last few years, I've also been involved a good bit with ozone therapy. And we've had some pretty interesting ways of doing ozone therapy. And that's what I'd like to talk about today. So let me get started with my slides. Okay, so let's see here. Now, I'm not sure what's going on. Let's see if we can pull them up again. I apologize for that. We thought we had this all squared away. There they go. Okay, so let's get a full view. So anyway, I'm part of the Institute of Regenerative Medicine, and basically you can see my websites down below and office numbers if you guys want any information about this. So we'll start. So basically what we have is what we call the EBO2 protocol, extracorporeal blood oxygenation and ozonation therapy. Another way we describe this, we call it the oxidative health protocol. And basically, we're incorporating medical ozone therapy, dialysis filtration, photomodulation, and a number of supplements in the battle against oxidative stress. And hopefully, this will promote longevity and a healthy lifespan. So it's not just living long, but it's living long and enjoying life at the same time. That's the real key here. Okay. So basically, as I stated, this kind of breaks it down a little more. We do ozone therapy, photomodulation, filtration, and then we use some other cellular therapies in addition to that, but we won't get into that in this lecture, such as V cells. So the goal of ozone therapy is to reduce inflammaging. This is a term that I believe was in Time magazine a few years ago, and it really is a very good description because we know that aging is tied into inflammation. So if you have inflammation, you're going to have aging and vice versa. And ozone is one method of kind of slowing this down. We can't necessarily turn back the clock of aging, but we can certainly slow it down. And again, we're going to go over some of the ways that we do that. So, when we talk about life and how we progress in life, it's basically we're on a balance board. And every day we walk on this balance board. It's basically antioxidants versus oxidative stress. We have oxidative stress here, we have antioxidants here. And we can see with the antioxidants, we have longevity, we have health, oxidative defense. And then with the oxidations or oxidants, we have age-related diseases, inflammation, abnormal problems, all sorts of things going on there. All right, so first we're going to talk about some of the other things here. Again, we see that as we kind of keep growing, we have certain oxidative defenses that start breaking down. And we get all sorts of bad things that happen. And we get membrane alterations, our lipid protein damages, our cytoskeleton damage. We get organelle dysfunction. Then we have organ dysfunction. And we have all sorts of things such as cancer, osteoporosis. Osteoporosis, by the way, we think is somewhat of an autoimmune disease. And again, a lot of these problems start at the mitochondria. The mitochondria, I'm convinced more and more, is the center of our health. If you have healthy mitochondria, you're going to be a healthy person. So basically, when we let's just get to the basics. How is ozone made? Well, we know ozone can be made by the use of electricity. For instance, sometimes you go out after a thunderstorm, you'll smell that pungent odor there, and that's ozone that has been made by the lightning. So you can see here, we get a molecule of oxygen, and then it gets zapped by the electricity. Then it kind of forms an extra an extra oxygen molecule, or I should say oxygen atom, and then it goes back and it forms a triple oxygen, and then it breaks down again. So it's a cycle that keeps going over and over. So here we have the cycle here. Again, just another slide that shows the same thing. Triple oxygen, then that oxygen breaks off. The single atomic oxygen disconnects from the ozone molecule, and it performs oxidation. Now, a very interesting concept, a very important concept. Ozone is considered a pro-drug. Now, what's the difference between a pro-drug and an active drug? Well, if I write a prescription for an active drug, you get that drug, and basically, as soon as you put it in the body, it starts doing things. Whereas a pro-drug, on the other hand, the drug is inactive before it's metabolized. It has to be metabolized in the body to become active. Whereas the active drug, basically, it's already active and it just starts affecting the body. So remember, ozone has to be converted in the body to become a pharmacologically active drug. This is a very important concept to keep in mind. Now, let's look at some of the peculiarities of ozone itself. First off, ozone is about 10 times more soluble than regular oxygen, so it readily dissolves in an aqueous environment such as plasma. So that's an interesting concept. Think about it now. You get a lot more ozone and, quote, oxygen in the plasma than you can with just plain oxygen alone. Now, there's a therapeutic window also of the amount of oxygen that can really have beneficial effects for you. And this is between 10 and 80 micrograms per milliliter, okay? That's per milliliter of blood. So basically, this is the magic window, the window that gives you some precise oxidative stress, okay? And that's an important thing. Now, another thing we'll get into more in the lecture, ozone generates two critical messengers. So it's not really ozone. As soon as ozone is entering your body, it's basically gone. It's converted into hydrogen peroxide, and it also converts into something called lipid oxidation products. We'll get into that in just a little bit. Now, the bottom line is the ozone effects, like other compounds, are determined by dosages. And this is not a very bizarre concept. This is a concept that permeates throughout all of medicine. And a really interesting way of looking at it, think about glucose. You know, if your glucose range in the blood is pretty much a normal range between 0.7 and 1 milligrams per milliliter, you're in good shape. But if that glucose goes down too much, you can have significant problems. You'll be in a hypoglycemic coma. And if it goes up too much, well, we know that that's a metabolic syndrome, diabetes, etc. Another thing I'll give you is oxygen. You know, oxygen is about 21% of our air. And basically that allows us to live many, many decades without a problem. But if we start breathing in pure oxygen, after a few days, it can be quite damaging to our body. So dose is very important in most drugs. Ozone is no different. Remember now, ozone is, like I say, a pro-drug. Now, hormesis, that's really what we're kind of talking about here. We look at a concentration of a certain drug. We can get a low dose, it's very beneficial, it's kind of the sweet spot. We start getting a higher dose, it becomes toxic, and it gets to the point where it can be very harmful and life-threatening. Ozone is the same as a lot of these other things. You know, we're looking at using ozone in this sweet spot right over here. We don't want this part over here, and certainly don't want to be over here. Now, ozone stimulates the cells to take action against both bacteria and viruses. Now, in Europe at least, there's been a lot of research and a lot of interesting papers that say ozone could very well handle most viruses and bacteria. They seem to have very good results using it against, for instance, COVID. And what it seems to do is destroy the lipid envelope around the virus, exposing its DNA and RNA. And once that happens, the virus is not long to live. A good article that I found recently from 2020, ozone is a natural bioactive molecule with antioxidant properties, and it's a new strategy in aging and neurodegenerative disorders. And I suggest you all read this. There's lots of articles, but this is one of the newer ones I found that really seems to work well. So now we're going to get to what I call the pillars of medical ozone therapy. These pillars are, you know, a couple of them. And one of them is optimizing oxygen utilization, very important. Okay, and we talk about NAD, and we'll get into that a little bit. Stimulation of what I call the NRF2 pathway and NQO1 pathways. Now remember, pathways, let's kind of talk a little bit about pathways. Think of your body, your organs and your cells as computer hardware. The pathways are the computer software. When you have a problem with your computer, usually the hardware is not a problem, it's the software. Same thing in your body. Most of the time, initially, the organs aren't the culprit. The culprit is the pathways that are controlling the organs and cells. Okay, so that's the thing. Now, another pillar of ozone therapy are ozone messengers, which I kind of mentioned, which I'll mention again and again and again. Hydrogen peroxide, reactive oxygen species, and lipid oxidation products. Another pillar of ozone therapy is we can remove debris from the blood. We can actually get stem cell stimulation by using ozone therapy. And last but not least is cytokine release and immune stimulation. So again, I kind of broached the subject a bit. The body's organs and cells are the computer hardware, and the pathways are the computer software. And speaking of pathways, These are kind of what I call the pathways of stem cell aging. And how stem cell ages, how your stem cells age is how you age. And this is basically the Rosetta Stone of anti-aging medicine. If you stimulate these pathways, you will stimulate anti-aging. And I want to draw your attention to just a few of these pathways. There's sirtuin pathways right here. What do? Mitochondria. Another very important one, AMPK. That's a nutrient sensing pathway, also involved with the mitochondria. They're all important, but those seem to have a little bit of extra importance. So we'll get into that in just a little bit. Now, ozone basically is responsible in many ways for ATP production. Now, ozone can accelerate glycolysis. Remember now, glycolysis is breaking down glucose into pyruvate and getting a very valuable hydrogen ion, free hydrogen. Okay, the free energy is released and it forms high energy ATP. ATP, as we all know, is the energy currency of the cell. And what this can also do is it can also form NADH. We know we have NAD and we have NADH. Now, the ratio of NAD to NADH is extremely important, and ozone can really help get that ratio in the right territory. You want that ratio to be about 700 to 1. Matter of fact, what we do is we look at NADH, and we consider that a marker of aging. The higher that NADH is, the more aging that patient is doing, the more probable oxidative stress he's having. Now, NADH is converted back into NAD, and this is another pathway, NQO1 pathway, which I which I mentioned already. So that's another pathway we keep in mind, okay? So one of the basic premises of ozone and ATP production is the formation of ozonides and their effect on the NAD-NADH ratio. So these ozonides come in contact with lipids and amino acids, and you can see what happens with that reaction. Ozonide plus NADH, what does it do? It produces harmless water and ATP, the energy currency, and it produces oxygen. So you're getting everything you need, basically. Some water, you're getting NAD, which is used for ATP production, and oxygen, which obviously we utilized also. Now, again, it's important when you're talking about ozone therapy, medical ozone therapy, you always have to mention NAD and NADH and how they work with each other. And you can see it again here. NADH gives up that H, and all of a sudden we get NAD, and this is basically the cycle of producing energy. Again, it's controlled more or less by NQO1 pathway. And again, we can see here the mitochondria, like I say, they're so important. And I just gave you this one little picture of the mitochondria. And again, hopefully my slides are available for you guys. The lecture is available. Go back and take a look at this at your leisure. I can't stress to you again, NAD-NADH ratio. As I stay here, NAD is a marker of aging, and therefore ozone can lead to anti-aging because it's diminishing the amount of NADH. But there's some other very significant aspects of ozone therapy that also contribute to anti-aging. And again, we can see here ozonides in the NQO1 pathway. It can lead to detoxification. It can lead to antioxidant production and P53 stability. Now, for those of you that aren't familiar with P53, P53 is called the tumor suppressor gene. And what this does, basically, it can analyze a cell. I believe the next slide may have this. Let me see if I can get to that. Let me see, we can go one more. Well, we'll get to that in a little bit. Basically, P53 can analyze a cell and determine if it should be something that should be kept and repaired or basically go on and perish. But I'll get to that a little bit later on in the lecture again. All right, so basically when we're looking at ozone, we have to realize, remember how I said earlier in my lecture, medications are dose related. Ozone is no different. If we have severe oxidative stress from ozone or anything else, it activates another pathway, a very important pathway called the NF-kappa beta pathway. Now, that's a pathway that basically is a thermostat of inflammation. It causes inflammation in the body. Now, sometimes this is a very important pathway to stimulate. You have a viral infection, you have a bacterial infection. This pathway is stimulated. It produces certain inflammatory cytokines, but basically it's designed to hopefully kill those offending organisms and get rid of them. We see this many times in COVID. Unfortunately, we've seen this in a cytokine storm where the body is in such a high gear of trying to deal with the COVID infection, it goes and stimulates the NF-kappa beta pathway to produce these inflammatory cytokines, which can cause significant damage. Now, the medical ozone that we're using is only causing moderate oxidative stress. This will basically stimulate the NRF2 pathway. I call that pathway the thermostat of anti-inflammation. It basically can control inflammation in a very important manner. So again, all dose related. Too much ozone, not going to help you. Too little, probably not going to help you either. And again, it's the tale of two pathways. We have the NF-kappa beta. It can do great things for inflammation when it's needed, but it's not going to help the body. Now, basically, when we have NRF2, it has a lot of good things for survival, growth, proliferation, and it can really make the difference many times. Oh, sorry about that. Okay, so again, another little slide showing the same thing. Now, NF-kappa, NRF2, for instance, we can see that hangs out in the cytoplasm. Now, given the proper push, so to speak, and for instance, we have hydrogen, remember I said hydrogen peroxide. As soon as ozone reacts with the body, hydrogen peroxide forms. Well, that peroxide then starts affecting this NRF2. Now, this is locked in place in the cytoplasm. Okay, now what that does is it lets the NRF2 escape. And when that NRF2 escapes from the cytoplasm, it goes into the nucleus. Now, NRF2 is really what we call a transcription factor. What does that mean in English? It means that it can affect certain genes, excuse me, to turn them on. And when these genes get turned on, what do? They start producing certain compounds and things like that. And what these genes do is they basically help make some very some very strong antioxidants. So basically, we have to remember one of the most important things in any medical ozone treatment is stimulation of the NRF2 pathway. It's the master regulator of antioxidant detoxification and cell defense gene expression. It regulates over 600 different genes. Now, NF-kappa beta, opposite. It's the master switch for inflammation. Each has their importance. We can't survive without either one of them working properly. And again, talking about the NRF2 pathway, we see that pathway. It's in the cytoplasm. It's kind of inert. It gets released, and once it gets released, it goes to the nucleus, and it forms what we call antioxidant response elements and genes, and basically it starts reducing inflammation dramatically. And again, we can see it here one more time. You have reactive oxygen species. It releases the NRF2, goes into the nucleus, and there we go. Certain genes are expressed, and guess what? Good things start to happen. So basically, some of the biological responses that are induced by ozone, well, we get increased levels of direct antioxidants. GSH is regenerated. You get, again, reduced levels of NADH, increased levels of certain enzymes. Cytokine-mediated inflammation is reduced, and a whole bunch of other things that go on there. Now, what's some of the relationships between, another thing we need to worry about a little bit, or not worry about, but just be aware of, something called heme oxygenase number 1. It's regulated by the NRF2 pathway, and it basically has a very important part in prevention of vascular inflammation. You have some kind of problem, what happens then? You'll basically get swelling. You'll get leakage from the veins. And that's what we were seeing a lot of times in bad COVID infections. These people would have all sorts of vascular problems that goes along with it, okay? So this is another important aspect of the ozone in that by stimulating NRF2, it's also stimulating this heme oxygenase, okay? Now, interestingly enough, it also helps release something called biliverdin and carbon monoxide. Now, interesting enough, you start hearing about carbon monoxide, well, that can't be too good. Again, let's kind of go back to the basics. In certain dosages, carbon monoxide is actually a very good stimulating molecule. It can really do a lot of good things. In higher doses, we know it's deadly. Okay, so we need to keep that in mind. Now, again, another influence of the NRF2 pathway, Goes back to the mitochondria. It basically influences mitochondria fatty acid oxidation. It supports the structure and functional integrity of the mitochondria. So anything that's going to stimulate the mitochondria in a positive way is great for longevity, great for your health. And that's what it does. Okay. Now, getting back to the P53. P53, again, is a tumor suppressor gene. What it does is it analyzes the cell, and I think I have it on the next slide now, if I'm not mistaken. Yeah, right here. So basically, P53 looks at a cell, it really analyzes it. And it says, you know what, you have minor DNA damage. I think what we'll do is we're going to fix you, and then we're going to let you go on and continue to divide and be on your happy way. Whereas if it looks at a cell and says, you have a lot of DNA damage, guess what? We're going to basically halt your division. We're going to basically help you perish. And that's what it does. It stops cell division. Now, the problem that we run into is that we don't have many copies of the P53 gene. And many times this P53 gene, over a period of time, can get mutated or damaged. Now, and that can lead to cancers. They think that a high percentage of cancers are because we have a mutation of the P53 gene. And another interesting fact about P53, if you think of an elephant, the largest mammal essentially known to man, it should be a setup for developing cancers because of its multitude of cells. Yet elephants almost never get a cancer. Why? Because they have multiple copies of the P53 gene. So ozone stimulates this, a very important anti-aging aspect of ozone. Now, how does ozone affect stem cell mobilization? You know, I've been a stem cell guy for 15 years. I mean, I've done thousands of cases. And I can tell you, ozone can make it work even better. Now, how does it really work? Well, basically, like a lot of things, it affects the bone marrow niche because most of your stem cells start there. Yes, indeed, adipose has a lot of stem cells, as do vessels, but there's a lot of stem cells in your marrow. And what it does is it produces some proteolytic enzymes, which are what we call matrix metalloproteases. And what these things do is they go into the marrow and they will go ahead, let's see if we have it on the next slide, and help free these stem cells by releasing them from their environment. And when those stem cells are released, they get into the circulation and go where they're needed. Okay, this is one of the actual bases of hyperbaric oxygen. It does a similar thing. Hyperbaric oxygen basically makes nitric oxide, and the nitric oxide then releases more stem cells from the marrow. And again, we can see that here, how the stem cells will just go into the vessels and go where they're needed, hopefully. So ozone can have a profound effect on release of stem cells from the marrow. So another way of thinking about it then. So pretty much this kind of sums up about the some of the clinical things about NRF2 and ozone, et cetera, and how important it is. Now, getting back to the ozone messengers. Again, we have basically the reactive ozone species and lipid oxidation products. Again, hydrogen peroxide, and then we also have the lipid oxidation products. So let's move on. And we can see that here. So we can see how ozone kind of has an effect. We have it on the plasma. We have the H2O2, hydrogen peroxide, affects the erythrocytes, leukocytes, and platelets, whereas lipid oxidation products are, affecting the endothelium, the bone marrow, and the organs. I want you guys to realize this also. H2O2 is kind of really fleeting momentarily. It doesn't really last that long. So its effects are more of a quick nature, whereas the lipid oxidation products can last for days and weeks even. And you can see what we have here. We can get improved O2 delivery, immune activation, and release of certain cytokines and things, whereas lipid oxidation products can release nitric oxide, release stem cells, upregulation of various antioxidant enzymes. So they both have their place. LOPs, basically long-acting, H2O2, very short-acting. And again, this is lipid oxidation products. They basically act on parenchymal cells, which is very important. Now, again, the lipid oxidation products trigger late effects, and they can also help upregulate the antioxidant enzymes such as SOD, glutathione, etc. Now, interesting, here's some other aspects of anti-aging and ozone therapy. Okay, so we can see here, here's ozone. These are polyunsaturated fatty acids. This is a cell membrane, which is very rich in these polyunsaturated fatty acids. Now, we can see here, this is something called the AMPK pathway. Remember I mentioned that when we were talking about some of the pathways. This is a very potent pathway of reducing aging, so to speak. And there's another pathway, the mTOR pathway. We're going to get into those shortly. So let's kind of go into that a little more now. AMPK, basically, it's a pathway that's critical for cellular homeostasis. It basically is a master switch to regulate cell functions such as uptake of glucose, burning of fats, and formation of new mitochondria. Again, we get back to mitochondria. We get back to glucose, things like that. It determines the body's fat composition. Medical ozone will stimulate this pathway, and it'll stimulate it very powerfully. Now, interestingly enough, some of you who may have done some intravenous ozone or something like that, you'll notice once in a while you say, wow, the patient kind of gets a little lightheaded, a little this or that. And we found the same thing, and then we started looking at some blood glucoses on these people, and the blood glucose was pretty low. And we realized that what happened is they were stimulating AMPK pathway. That pathway is driving the glucose into the cells, and the blood glucose is really dropping down. So this is one of the reasons why you have to have your patients, with a good amount of glucose in them. Make sure they eat something before you're doing any medical ozone, or IV ozone, I should say, because that's very important, because otherwise their blood sugar is going to drop. So just keep that in mind. Very important aspect. And we can see all the various things that AMPK is associated with: glucose uptake, fatty acid uptake, mitochondrial biogenesis, there we go again, fatty acid synthesis, cholesterol, lipogenic enzymes. It does so many different things. Now, what else the ozone gets involved with indirectly is it basically helps prevent activation of the mTOR pathway, okay? mTOR pathway is another interesting pathway in that it actually helps with cell growth, cell division, etc. But it's really not good for longevity, because basically what it does is it really revs up the growth so much that it causes aging. So if you can block this pathway, then you're going to basically do some good things for anti-aging. And the AMPK will block this pathway. So by ozone stimulating AMPK, it's blocking this pathway. mTOR means mechanism target of rapamycin. Now, rapamycin is an interesting compound. It's a compound that was basically found in the dirt of Easter Island. It's thought to be a very potent immune suppressor. So they give it to patients who have been on, who've got an organ transplant, let's say, or something like that. But now there's a lot of studies afoot that show that this may be something that's a very potent thing for anti-aging, because it blocks this pathway. So that's something to keep in mind. And again, we can see all the things here. You get aerobic glycolysis, you get RNA translation, so you get proteins, membranes, all good things if you're trying to repair something. But if you're trying to slow down aging, very bad things. It's like, live fast and die young. That's basically what this pathway does. Now, we can't have it totally blocked, because obviously that's not good, but we need it to be functional somewhat. Now, another thing that this pathway blocks, interestingly enough, is autophagy, where we basically recycle certain parts of cells and things like that. Now, that's a very important thing, again, for anti-aging. Autophagy is extremely important, and you can see that right here, okay? So basically, if we stimulate autophagy, we stimulate longevity. No ifs, ands, or buts about it, okay? Now, again, let's talk about some of the hydrogen peroxide aspects of ozone therapy. Right here, nitric oxide is produced, but look at this, carbon monoxide. Again, like I said, in very small doses, carbon monoxide is a very important stimulating molecule. And we can see that right here, okay? It has diverse functions, including modulation of neural signals, inflammation, cell proliferation, and cell death, and smooth muscle contractility. So it's it's an emerging field now, very important signaling molecule. Again, we know how deadly this is in a higher dose. Not just bad, it's just deadly. But in small doses, it's a very potent stimulator of certain effects. And we can see that again here in the ozone systemic effects on the platelets, release of certain growth factors. This is VEGF, that's a growth factor that's very important in formation of blood vessels. Platelet-derived growth factor, important for tissue repair, as is TGF-1 beta. It blocks inflammation, good for wound healing, all kinds of good things there. And again, hydrogen peroxide, our buddy. Now, can ozone therapy produce a significant improvement in blood flow and oxygenation? Absolutely. It does it by a couple of different methods. Remember, now we have nitric oxide, which causes vasodilation. We have carbon monoxide, which can also do that, and various release of various growth factors that can help increase the blood supply. Now, interesting thing, what we're also doing is we know that ozone can do something, form heat shock proteins. Now, we're also doing this with photomodulation, which I'll get into in just a bit. But basically, heat shock proteins are a family of proteins that are in response to an exposure to stressful conditions. Now, what heat shock proteins really do, when all is said and done, is they help a protein fold properly into the cell. A lot of diseases are basically related to improper protein folding. And this kind of gives you a little cartoon of what happens. So basically, we have a stress, we have a damaged protein, and then the heat shock proteins come into play. Basically, they can repair, reactivate the protein, or proteases will go ahead and basically do away with that protein, because if it stays, it's going to cause damage to the cell. So heat shock proteins are very important, and ozone does stimulate them. And the primary role of heat shock proteins is to keep order. It's to get that protein to fold properly back into the cell. But another job it does is activate immune responses. So that's another important aspect. And again, think of a heat shock protein as kind of like a chaperone. It's like a little helicopter hovering over these proteins, making sure they do things the right way. And if there's the first sign of bad behavior, they'll basically go ahead and see if they can prevent that. Okay, now let's get to the actual EBO2 protocol. It's called the Oxidative Health System Protocol, whatever you wish to call it, doesn't really matter. But basically what we're doing is we're taking almost the patient's entire blood supply, 5 to 7 liters, over a period of about 60 minutes or so. And we're running it through, like I say, the filtration with a dialysis filter. And at the same time, we're removing impurities with the dialysis filter, such as cholesterol, some heavy metals and toxins, some petroleum products and things like that. We're also removing something called beta-2 microglobulin, and I'll get into that in just a little bit. Now, there's a big difference between the procedure of EBO2 and autohemotherapy. Autohemotherapy, some people refer to it as a 10th pass technique, where they'll take maybe 200 cc's of blood at a time. It's removed, it's placed in an IV bag. They inject it with the heparin, and then they inject it with ozone gas, kind of mix it up, then take that blood back out and inject it back into the vein. That is kind of old technology. It's not really the new stuff, so to speak, because there are a couple of things that's going on here. You're not really getting any filtration or anything. You're depending now on your kidneys and your liver to, get out some of the bad stuff, which they can and sometimes can't do. Also, you're getting basically hemolysis of a lot of red blood cells, because when you start shaking these things around and taking them out of the body, they're going to react by basically lysing. So you're going to get that done. And there's also an increased chance of contamination. So basically, this slide, it kind of, just shows you basically what they're doing here. So what we have here is we have a peristaltic pump. So what does that mean? So we have an IV, this IV, let's say, is coming from the patient, and the pump is actually sucking the blood out of the vein. So we're able to get a lot more blood flow, and then it's pushing it through, okay? So that's the peristaltic pump aspect of it. Now we see our dialysis filter right here. So the blood would be entering from below, okay, and then it kind of travels up the filter, then it gets hit with ozone gas here, then the blood goes over here, and then it kind of goes out into the chamber and goes from there. Now we can see the apparatus as it's set up. So here we are here. This is the blood going from the patient, going into the dialysis filter. and it's getting hit with ozone gas. And look at the difference in the color of blood. Look at how dark this is. It's hard to really maybe appreciate on the slide, but this is pretty dark blood, venous blood. Now look at the blood after it's hit with the ozone, much, more vibrant red color. So basically, we're removing a lot of toxic blood products. We're hopefully getting rid of some petroleum byproducts, inorganic substances. It's interesting because we have a fairly big vessel, I should say, catheter that we're using. We see a lot of fat microparticles that are coming out also when we do this, and they're being removed by the filtration. So it's pretty impressive, and the patients are very impressed when they see this. Now, another added bonus is that we're removing something called a beta-2 macroglobulin. And it's a very sneaky compound, and you can do a lot of reading about this, but basically it seems to be produced by the cells of the immune system. And it appears as a foam in the container. And let me see if I have one that shows you. Well, here's some more information on it. And we can go on to this if we have to, but I think we can just have you read the slides more, and you can see basically it's pro-inflammatory cytokines that are responsible for this. And again, it comes from the immune system cells. Some people say that beta-2 is a marker for disease. Some people say that it's actually associated and can help cause disease. But this is what we get out of it. This is a container from someone who is getting a process. And you can see the size. I mean, there's someone's hands. And this is all filled with this foam. Now, I have a lot of people, they'll get almost nothing in there. They'll just have a little sliver of foam and some water in there. But this is someone who probably was some kind of autoimmune disease or some kind of chronic infection. And we can see it. We'll see that. And then the first time it's very, very prevalent. And then as we start doing more treatments, it really goes down to not much of anything. So, there's a saying that elevated serum beta-2 microglobulin basically is associated with an all-risk cause of death. So it's an important thing to remove from that circulatory system, so to speak. And again, some more things. It's about the pro-inflammatory cytokines. Not a good thing to have floating around in your body. And we're removing it with this protocol. So a lot of it is from the platelets itself, which seem to drive inflammatory diseases, and then it induces pro-inflammatory monocytes. So again, it has a pretty significant effect on our immune system. And basically, it also seems to be affected by advanced glycation end products. Basically, when you have sugars and you really basically break them down, or sometimes you can see that when you get a piece of something that has sugar in it, or even a piece of meat, and you can cook it, and you'll see how it has that coating on it, so to speak. So these are some final thoughts on the beta-2 microglobulin. It involves a network of cytokines modulating the development of several cell lines. So if you can diminish that, you're probably going to also help reduce inflammation. Now here we have the safety chamber, again, going on our EBO2 machine, and basically prevents problems of air embolus, because you'll have your technician. We always have one of our nurses or technicians with the patient at all times. We don't want to all of a sudden start feeding air into his vessels or something like that. And knock on wood, we've not seen any problems whatsoever with this. Now, photomodulation. I'm always a big fan of photomodulation. For many years, I've been using it for all my cells, be it PRP, be it fat, be it bone marrow. I always use photomodulation. And we use different wavelengths of light to modify the behavior of the cells. So in this case, we're using a UVA, which is a wavelength of about 370 nanometers, and we're using a red light 660 nanometers. And again, this just gives you an idea of some of the mechanisms of light therapy. And again, it's light therapy. It's photonic therapy. It's not heat, okay? When you're using a laser and that laser is getting something hot, you're not really getting that much energy out of it. It's not really doing that much good for your patient or anything like that. So basically what our red light sleeve does is we can help produce more ATP, make the mitochondria work better. So mitochondria are basically, wonderful organisms. They make energy, but they also have a lot to do with the various internal workings of the cell. They also help release nitric oxide, which is a good vasodilator and things like that. And you can see what we've developed here is this is the line that's going back to the patient. This is a light sleeve, a photonic sleeve. It's about 1 foot long, and we're hitting it with all these red lights, and there's all sorts of mirrors in here. Everything is kind of mirrored. And then the UV light, UVA. We don't use UVB. We can get it if somebody really wants it. But UVA, we like to do it because it's a heat stress. Excuse me, it's a stressor, and it helps produce more of the heat shock proteins. We're not using it necessarily to kill organisms. UVB might be able to do that, but UVA, we're doing it for the heat shock purposes and things like that. And that's what that part looks like. Now, it's interesting. I took this with my phone, and if I look at this with my naked eye, it certainly does not seem as blue because my eye cannot pick up the colors like my phone can. And this is basically the setup here. So we have one line going from the patient, it's going into the UVA sleeve, and then the peristaltic pump gets it, pumps it into the dialysis filter, gets hit with ozone here, then it goes out this way and goes all the way around and going back to the patient. And we're looking at some other things that, maybe we might start using some blue light because we have that already, the UVA. It seems to increase exosome production from umbilical cord cells, so we figured probably would do the same thing with regular stem cells too. Remember, there are stem cells floating in your circulatory system. Now, can supplements increase this effect? You betcha. Okay, these are some of the things that we use. Terstilbin, we know that really is important for the sirtuin genes, or sirtuin proteins, so to speak. True Broc is a broccoli extract. Turmeric, Ultra Cure, good one. Green tea extract, resveratrol. Fumaric acid, I've helped just design recently a NRF2 pathway stimulator, and fumaric acid is part of it. Now, fumaric acid is a very potent stimulator of this, and the FDA just recently released a compound for unrelenting multiple sclerosis, and the main component of it is fumaric acid. Quercetin is also very good, NAD, and not NAD itself because you're basically going to get the precursors of that, and then of course nitric oxide. So basically, what are we? Again, we're trying to get that NRF2. That's the real key. If I have to say one important thing about medical ozone, NRF2 pathway activation. That's probably the most important aspect of all the activation things. Is there any side effects with the EBO2 protocol? Again, occasionally somebody may get chest tightness, and those of you that have used intravenous NAD, you'll find it's the same exact thing. And what happens is, remember now, the patient is getting NAD produced from the EBO2 protocol. So what we find is if you give the patient some trimethylglycine ahead of time, like we do when we do NAD, they will not have any chest pain or anything like that. It'll go away. Basically, it's the TMG is a methylation agent. It works very well. I already mentioned to you how the patient has to make sure that they've eaten properly, have enough glucose on board, because that glucose is going to be driven into the cells. So that's an important aspect also. Another thing that we use, these are very important aspects of our treatment, mainly for stem cells. These are growth factor patches. Now, basically, they are registered with the FDA as an OTC, but we're using them off-label. For instance, the OA patch is basically, it's a clone of what the stem cells make. It's basically IGF-1 in a patch with penetrating molecules. AI is interleukin-10 and interleukin-1 antagonist. And RG is basically fibroblast growth factor 1 with some IGF-1, et cetera. What I like to use is AI. What we'll do is we'll take this, we'll put it on a tegaderm patch, we'll put it over the carotid arteries, and we don't get anything like a Hertzheimer reaction. Pretty much goes away because the patient is basically getting buffered by these patches. So they work pretty good. Now, what we're looking at in the future is a couple of different things. Looking at maybe utilizing hydrogen gas and xenon gas. Xenon gas, I'm very interested in. Believe it or not, xenon gas is not allowed by the World Anti-Doping Agency because it's a significant stimulator of performance enhancement. What xenon gas does is it will actually make the patient, it will stimulate his production of erythropoietin, so he'll have much higher blood counts. And it does a lot of interesting things. We're looking into that, not for the professional athlete because that would be a violation, but for general well-being. Looking at a UVA, or excuse me, a UVC or UVB strip. And we're also looking into magnetic stimulation of the blood. You know, blood basically Oxygen is magnetic. Hemoglobin's, the iron is magnetic. So we're looking into that. I'm not sure if we're going to do anything, but there's some of the things about molecular hydrogen and xenon. And with that, I think we are done. I'm more than happy to answer any questions. So I don't know how we go about doing that, if I can see you or hear you live or what exactly.